When it comes to discussions of human development, there are several terms that are often used. Oocyte is the female germ or sex cell that is produced in the ovaries. When the oocyte has matured, it may be called a secondary oocyte or matured oocyte. Spermatozoon, the plural sperm, refers to the male germ cell which is produced in the testes. Several sperms are expelled from the male urethra during ejaculation. Zygote refers to the cell after which an oocyte and a sperm unionize after fertilization, which takes place in the oviduct of the female. Now it is difficult to determine exactly when fertilization occurs because the process cannot be observed in vivo. So physicians calculate the age of the embryo from the first day of the last normal menstrual period, LNMP. This is the gestational age, which is 2 weeks longer than fertilization age. This is because the oocyte is not fertilized until about 2 weeks after the preceding menstruation. So when a pregnant female enters her physician's office and given an age of her fetus, 2 weeks must be deducted to determine the actual fertilization age of the developing human. Cleavage refers to the series of mitotic cell divisions in the zygote that result in the formation of the early embryonic cells, or blastomeres. Medulla is a solid mass of about 12 to 13 blastomeres and is formed by cleavage of a zygote. The blastomeres change shape. They align themselves tightly against each other to form a compact ball of cells. This compaction is probably mediated by cell surface adhesion, glycoproteins. In about 3 to 4 days after fertilization, the morula stage occurs. This is just as the early embryo enters the uterus. Just a little tidbit, the morula was given the name because of the resemblance to the fruit mulberry or blackberry bush. In Latin, the term morula is moris, which means mulberry, hence the name morula. Once the morula enters the uterus from the fallopian tube, the blastocystic cavity is fluid-filled, which develops inside it. This change converts the morula into a blastocyst. The inner cell mass in the blastocyst is the beginning of the embryo. Implantation is the process during which the blastocyst attached to the mucous membrane, the lining of the uterus, or what's referred to as the endometrium. The time between fertilization and the beginning of implantation is called the pre-implantation period of embryonic development. This is a 6-day period. During gastrulation, the formation of a blastomere into a gastrula, a 3-layer or trilamellar embryonic disc forms. This is done about the 3rd week after fertilization. The trilamellar cell layers will differentiate into the various tissues and organs of the embryo. The slide we are looking at illustrates a couple of terminologies we have discussed previously. The first drawing on the top left-hand corner of the screen shows the development of a follicle cell. In the center of the cell, you will see what's referred to as an atrium, which contains follicular fluid. A bit south of the atrium, we can see the oocyte with the nucleus in it. The primary oocyte is pushed to one side of the follicle, where it is surrounded by a mound of follicular cells, the cannulus oophorus, which projects into the atrium. Looking at the top right-hand corner of the screen, we can see the sperm penetrating its way through an oocyte. The first picture illustrates the sperm making contact with the jellycoat of an egg. Here we see the acrosomal membrane of the sperm break down. The acrosomal enzymes digest the jellycoat of the cell. Actin filaments polymerize, forming a process. Receptors on the volatile envelope bind to a species-specific glycoprotein on the sperm. At the lower bottom of the screen, we see the several divisions of a zygote. Illustration A paints us a picture of an egg right after fertilization, when the cell becomes 2. Picture B is when 2 cells become 4, 4 become 8, 8 becomes 16, 16 becomes 32, which we know from terminology as a morula. Directly to the right of the zygote divisions are illustrations continuing the growth and development of the morula. 2, again, is what we call a blastocyst. Now let's continue with the addition of terminology. Embryo, the developing human during its early stages of development. The embryonic period extends to the end of the 8 weeks, by which time the beginnings of all major structures are present. Physicians can measure the size of the embryo using the CRL method, the crown-rump length, measuring the vortex of the cranium, the crown of the head, to the rump, or the buttock. Early embryonic development is described in stages because of the development of certain morphological characteristics. Stage 1 begins at fertilization and ends with stage 23, which occurs on day 56. The fetal period begins on day 57 and ends when the fetus is completely outside the body of the mother. Conceptus is the term used to describe the embryo and its annexa, which is appendage or adjunct parts of the embryo. This includes the placenta, the amnion, chorionic sac, and yolk sac. Primordium is the beginning of the first discernible indication of an organ or structure. For example, the primordium of the upper limb appears as a bud on day 26. The terms annulage and rudiment have similar meanings.